This is the general.
Okay, Sunday morning, 11 o'clock means it's time for chai and wine. So thank you all of you who are here. Thank you those who are watching online. Third Sunday, we are at Rupal College. And in case anybody is watching this for the first time, Chai and Wine is an outreach offering from the IFR, where we are at Prithvi Theatre, Mumbai on the first Sunday of the month. The third Sunday, we are at Rupal College in Matunga. And if a month is the fifth Sunday, then we are live from a lab in IFR. That's the only session which is online only. The ones at Prithvi and Rupal are both in person and online. Before I get to today's session, just in case you would like to know, March indeed has a fifth Sunday. 31st March is a Sunday. We will be giving you a live tour of the low temperature facility at the IFR on Sunday, 31st March. Following that, the first Sunday of April at Prithvi Theatre, we will be looking at the world of chemistry, or more like an interface of biology and chemistry, where we have a topic on how one can look at fat molecules or lipids inside living cells. So that's coming up. Those are the next two sessions. The next one at Rupal College, we will be posting in a couple of days. Always follow our website to get the updated details. So today, we're going to be looking at cells. And to tell us about them is Badal Singh Chauhan. Badal is a PhD student at the IFR in the Department of Biological Sciences, where his research focuses on understanding how things move, I think in nerve cells, in tiny little worms or something. I hope you will tell us about it later. Badal did his master's at Aligarh Muslim University and then came to TIFR to do his PhD. So without further ado, let me hand it over to Badal. You can share your screen so that the online audience knows what's going on. Online audience, if you're watching us, please remember that the only way we can get your questions is through the chat. So please put your questions and comments in the chat. We have people monitoring the YouTube channel, and we will look at them and take them during the talk. So please put your questions and comments in the chat. And of course, people who are here, you can raise your hand and ask questions whenever you want. So yeah, on that, why don't you share screen and go ahead.
Thank you, Anna, for the introduction. I'm going to share my screen.
Go to board, three dots, and height floating meeting controls. Yeah. Good.
Okay.
I'll just move it up. There it is. Good.
Okay. Thank you, Anna, for the introduction. I'm Badal. I'm doing PhD at TIFR Mumbai. Okay. Okay. So I'm sure every one of you has seen a factory. Either you have visited or you have seen in the movies or some documentary, that where the things are packed, things are processed and sent to the certain distances. And the same thing, the factory kind of thing is happening in your body in billions of cells. So in my talk, I'm going to talk about some processes which are happening in the cell in my talk. It is not. Yeah. Okay. So I'm sure everyone has seen a building. All the kinds of buildings are made up of small bricks. So there are certain other things which helps in the connecting of these bricks, but there is a basic building material, which are the bricks. In our body, all the living organisms are made up of cells. Cells, a very small insect to a large water animal, to a water plant, to a large banyan tree. All the things are made up of the cells. Animals can be, organisms can be unicellular. They are made up of only one cell, or they are like us, multicellular also. So to understand the there are different compartments in the cell. So this slide seems slightly congested, but I'll go back through. So like a factory, there are things which control what is happening inside the factory. It's like there is some controlling room. Same in our cell, there is a nucleus which control all the processes which are happening in the cell. Like factory unit power, mitochondria provides energy to the cell. There are some places where, like conveyor belts and other things, where you made a product, packaged and sent to the other directions. These things are done by endoplasmic reticulum and Golgi apparatus. There are things you need to store in the factory. Same thing, cells also need some space, so vacuoles. There are some places you need to digest waste product and other things, so you need lysosomes. This lysosomes. And of course, to protect your factory, you have an outer wall. Same, and from that outer wall, only certain things can come and certain things can come out. Same in the this whole cell is protected by a cell membrane. Only certain things can come out and go out depending upon what kind of the cell is. Now, to understand what the cell is made up of. So the food we eat has these components like lipids, carbohydrates, and proteins. Lipids are the fats. We generally call them as a fat. Plasma membrane and other things, all the membrane which is present on the cell is made up of lipid, majorly lipid. Nucleus, nucleic acids. In a cell, there is a genetic material. Most of us heard about the DNA, which contain the genetic information. So this is made up by nucleic acids. Carbohydrates, main function is to provide the energy. They provide energy, but they also have some role in formation of these lipids and other things also. And last is the protein. So protein are the major building blocks of a body or a cell. So in my next slide, I'm majorly focus on what are the proteins and how they function, how they transport in different parts of the cells. So first, let's introduce what are the proteins. So proteins are made up of amino acids. So the whole body is made up of different building blocks. Same, the protein is made up of long chain of amino acids. So amino acids are small biomolecules. They have carboxylic group and they have amine group. These different amino acids. So in the cell, there are 20 different kinds of amino acids. They can arrange in any order. So one amino acid is attached with another amino acid, and then another amino acid attached with the next amino acid. So each of 20 amino acids can attach with any other amino acids in any arrangement. Same, you imagine you are like they are arranging in a string. So you are holding your friend hand and your friend is holding your another friend hand. So these kind of structures are the same. They make a long chain of amino acids, and on the basis of what kind of amino acids and in what arrangement they are present, that decides the function of a protein. So now the proteins are formed. So proteins are majorly formed in this endoplasmic reticulum, rough endoplasmic reticulum. You can see different compartments. So similarly, in the factory, there are different compartments, right? In the cell also, there are different large number of compartments. So how the proteins are transported from one place to another place, that we'll discuss in next few slides. So majorly where proteins are synthesized in rough endoplasmic reticulum, they goes to Golgi. Here the decision is taking place whether they want to go out from the plasma membrane, whether they go to other compartment, and from their decision also taken in the other compartment where they want to go out, they want to go to the lysosome. So we'll discuss how it is happening. So a big question is, cells have large number of proteins. The number of proteins are in thousands in numbers. So how you decide a protein in a tiny cell, how it reaches from one compartment to another compartment? I'm sure everyone has sent something to their friends, so while writing the address, we write the pin codes. Pin code decides the place where the parcel is going to be sent, right? Same, the pin code has some numbers. Let's say first decide about the zone. So India is divided into 4 different zones. So this decides the zone, and then subzone, and then sorting district, final district, and then on the basis of last 3 number, which post office in last they has to go. Similar kind of mechanism happening in the cell also, where the protein after coming, is when the protein is forming in the rough endoplasmic reticulum. Proteins have some signal sequence. So signal sequence are some amino acid signals which decide the destination of this protein. So after forming in the ribosome, let's say the protein is forming in the cell, if they have the signal sequence for targeting to the endoplasmic reticulum, what this machinery will detect that now the protein is coming, it has this ER targeted signal. So in simplicity, I'm using the word endoplasmic reticulum, but that compartment can any compartment if the signal sequence is there. So if the protein has ER targeted sequence, what will happen is this whole complex will bind and then the protein now enter into the endoplasmic reticulum. Next is from the ER, proteins comes to the Golgi. Also, there are certain signal sequences which helps in sending proteins from Golgi from ER to the Golgi. Now, another round of decision is taking place in this Golgi compartment. If the proteins has a sorting signal destined for endoplasmic, for outside the plasma membrane, protein will go out to the plasma membrane. If they have a signal which require it to send to the lysosome, protein will go to the lysosome. Let's say here the protein has a sorting signal or signal to send it to the endoplasmic reticulum. Let's say the protein does not have. What will happen? Protein will remain in the cell. It will not go into this endoplasmic reticulum. Now, on the basis of, so protein is present in the cell, in the cytoplasm, floating around. Now, on the basis of what targeting signal it has, if it has a targeting signal for periosomes, it will go into the, if it has a targeted signal for chloroplast, the protein will enter into the chloroplast. It is present in the plant cell. So chlorophyll, it has chlorophyll which helps in the photosynthesis. If the protein has a targeted signal for mitochondria, it will enter into the mitochondria. And if a protein has targeted signal towards the nucleus, protein will enter into the nucleus. So what decides so to summarize the whole slide, the target of the protein is depend on the signal sequence which the proteins have. Like, so just for simplicity, I'm just showing the different targeted signal. Let's say KDEL. These are 4 amino acids. If these 4 amino acids are present in any protein, the protein will remain in the endoplasmic reticulum. If these sequence is present, the protein will send it to the nucleus. So actually, while doing research, if we want to artificially target to a certain protein into the nucleus, what should I do? I should just tag the protein with these amino acids. What will happen? Now the protein will reach into the nucleus. Same thing I can do with mitochondria and lysosomes. In most of the cases, the signal sequence is made up of amino acids or protein, but there are some other things also present. Let's say mannose 6-phosphate. So mannose is a kind of carbohydrate, and it is present on any protein. If this mannose 6-phosphate tag is present on any protein, now the protein will end up in the lysosomes.
Let's say this whatever PKLKRU.
Yes.
If it is there, it just means that
it will go to the.
That is not there in anything else. That is very big.
So certain sequences are unique to the nucleus. So this is one of the sequence which is unique to the nucleus. Yeah.
But it's not that is required to the protein something else.
Only to the targeted to that certain compartment. Yes. Is
it present in a particular part of the protein?
So it can present mostly the present. So let's say if the protein is on the secretory pathway, the protein signal is present on the C terminal of the sequence. Let's say ER targeted signal. It should present in the early steps, because after the protein is translated, maybe it will not enter the ER. So this kind of signal sequence should present in the starting of the protein. So I spoke about how these signal helps in sending the protein to a different compartment. Now I'll just discuss about how this machinery works, how they detect and sense the how they sort out the protein and send to the compartment. Let's say there is a donor compartment. You can assume it as a endoplasmic reticulum, Golgi, or any compartment where the proteins are present. Now I want to send a certain sets of protein to a compartment where they needs to be sent. So these are the proteins. Some are transmembrane proteins. They are present on the membrane, and some are present inside the vesicle or inside the compartment. What will happen is they are recognized by the cargo adapters. So what is the use of sequence? You should have a certain things to detect the sequence, right? You should have some proteins that detect the specific sequence. So these are the cargo adapters. What they do is they come on the membrane of that compartment. What they do is they detect, they sense what is the signal sequence present on the protein. If the signal sequence is matched with the cargo adapter, now the cargo adapter and the protein will bind. So you can see this cargo adapter can bind with this, can interact with this protein. Now they are coming together, and after some time, lot of cargo adapters come and it starts forming a vesicle. After some time, vesicles pinch off from the compartment and then release. So this is one kind of regulation where you decide which protein needs to send. Another regulation is, let's say in a factory, your parcel is made, you send somewhere, but how to decide from which compartment it should accept? Because now this vesicle is a small vesicle, you can see it has a different proteins. It can go anywhere, but so in the cell it doesn't happen. It doesn't happen because of some other protein. So when I'm sure most of you receive some parcel from Amazon and Flipkart, when the person come, they ask you the OTP. Tell me the OTP which you have, and you say it's 1234. The person also check in his phone. Oh, the OTP is matching 1234. On the basis of that, the person delivery associate hand over the parcel to you. Similar kind of thing happening in the cell also. So whenever a new vesicle is forming, which have the proteins, they have some snare complexes. So you not need to remember the names, but there are certain proteins which helps in identifying the compartment or fuse with the compartment where it is destined. Let's say, so there are two different kinds of vesicles are forming in the membrane. It has one kind of V snare. It has another kind of T snare. So this T snare, which is present on the donor compartment, and another T snare is present on the acceptor compartment. If only they are interacting and they can match, only the vesicle can come and fuse with the compartment. Understand? So let's say only these two can interact with T snare. This V snare cannot bind with this. So even if large number of vesicles are present here, they will not fuse. Fusion only happens when both the snares present on this compartment interact together. So this is another kind of regulation, how the proteins is destined towards a specific compartment. First is at the type of biogenesis, you sort out all the proteins. Second regulation happens at the time of fusion. Only fusion happens to the specific compartment, and that is done by the certain proteins which are present on the vesicle as well as the compartment. So okay. So the question is, what decides if the protein is end up in the endoplasmic reticulum? It is the protein sequence. The sequence, it is the amino acid sequence which are present on the protein. So if it has ER targeted signal, I can go back. If the protein has ER targeted signal, so there are different machinery complexes. So what can happen is in the cytoplasm, these complex can work. Sorry. Oh, in the cytoplasm, what happens is there are different machinery which are present. If they detect that now the newly synthesized proteins which is coming out after this ribosome, if they have a signal which is required to attach with the ribosome, the proteins comes into that. If does not happen, they will form complete, they did their complete biogenesis in the cytoplasm. So it's the signal which decides. Yeah. Okay. So this is the summary of simple steps which are happening in the cell. Now I'm going to walk through is what we yes. Yes. Oh, okay, okay. So here there are there are different compartments. So for example, there are two different compartments. Upper one. Okay. So for in this vesicle, they have these snare proteins. They can bind with these T snares only. So compatibility, right? I have let's say you have a vesicle, you have a protein that can interact with another compartment which have this kind of a structure. The protein, when they come and they are both are compatible when both the snares are present, which the new vesicles which is coming and the vesicles which are in the donor acceptor compartment. When they are compatible, only in that condition they will fuse and then they will give the protein. If they are not wherever it can attach, it attaches. The one which cannot attach, it is not capable. Yes, so in many cases, in many cases it happens, but not most in the cases. Let's say if you want to send something to so there is not only a regulation which decides which compartment needs to bind. Let's say you want to send a protein to synapse. You want to send a protein which to the synapse. So first regulation may come from you are directing towards either based on the motor or other vesicles. The vesicle itself first going into this compartment. Now the vesicle have come in the close proximity with other things and then fuse. So regulation, it's not always the vesicles are just randomly forming and then doing diffusion in the whole cell and then fusing, but there are other kind of regulations also based on the directed transport. Yes. Typical time scale, it depends actually. But fusion, after forming fusion happens very fast actually. Very, very fast, less than a second. If that compatibility is there, it happens very fast. Same happens in the synapses actually. When the action potential come, within a fraction of microseconds, complex form vesicle fuse release its content. So what we do in our lab. So I was talking about the cells. We works in a specialized cells. These are neurons. You know, most of us know the function of a neuron. In our brain, there are what we are seeing, thinking, we are feeling. Everything is regulated by the neuron. So we work the neurons. It's a typical diagram of a neuron. It has a long, small structures. These are dendrites, which sends the information surrounding. There are cell bodies which regulate the process of the whole neuron. There is a long tubular structure which is axon. And now one neuron is connected to another neuron by two ways, but one way is the chemical synapses. So it's a typical chemical synapse. And the job of neuron is to either process the signal and send signals to the next cell. How they do this? One way by doing with this chemical synapse. What happens when a action potential comes? There are a lot of vesicles present at the synapse. They have the vesicles have a small chemical substance. These are called neurotransmitters. These neurotransmitters are filled inside a vesicle. So so glutamate is one neurotransmitter, very small amino acid. These neurotransmitters are filled and packaged inside the vesicles. So when the neuron is not firing, neuron is in a resting state, what happens is just these vesicles are in the close proximity at the presynaptic region. This is one neuron, next is another neuron. So when the action potentials come, what will happen is some channels present on the plasma membrane are active, some calcium and other ions increases, due to which these vesicles now fuse with this plasma membrane. When the vesicle fuses with the plasma membrane, what will happen is the content it released. Now this small molecules or neurotransmitter release, now it sends signals to the next cell. So this is just a schematic representation of the neuron and how the chemical synapses work. So in our lab, what we are interested in mainly question related to the biogenesis of these synaptic vesicles. So how these synaptic small synaptic vesicles form in the cell body, how they transported, how these are regulated, how their composition is regulated. So all these questions we are interested in. Now, if we want to understand all these things, how can I understand? I need a model organism. I cannot take neurons from anyone, like, please give me a few neurons I want to study. That I can't do. I need a model system where I can use the neurons to study all these processes. So we are using C. elegans as a model organism. C. elegans is a small 1 millimeter worm, so you cannot see with the naked eyes. It's a very small worm. It was first isolated start using by Sidney Braden 60 years back. He got Nobel Prize in this also. So why we are using C. elegans as a model organism? It has a simple nervous system. We have billions of neurons, but in C. elegans, all the neurons are characterized. So it has only limited number of neurons. The number is 302. Only 302 neurons are present, and we know in most of the cases which neuron is connected with which another neuron. So we know whole the connectome, which neuron is connecting with it. So hermaphrodite, yes. In hermaphrodite, in wild type condition, yes. In wild type condition, yes. 1 millimeter. Very small, very small. Yeah. So it's a very simple nervous system. We know which neuron interact with what, where is the neuron. We know the kind of neuron, more and the function of neurons also in most of the cases. So we have a very simple nervous system and we understand it. Second is it's very small, so we can easily grow in the laboratory conditions. It has a very short lifespan. So if I want to do experiment, we should do it very fast. We should not wait for 6 months just to grow that organism and check what is happening. So it's a very short lifespan. In few days, we can get the next progeny, and lifespan is around few weeks. Few weeks, 3 to 4 weeks, yes. And it has a transparent body. I'll tell why it is important in our case. Let's say we want to see a neuron, and we see a neuron with the help of microscopes. And let's say if an animal is opaque, we cannot see with the help of microscope. So what C. elegans body is completely transparent. It has conserved process also. So we want to use a model organism where we understand the process using C. elegans model organism, and then we can say that these kind of processes are happening in human neurons also. So many of these processes are conserved in C. elegans as well as human. So that's why. And last point is easy manipulation. So it's a very small generation time. We can do small manipulations in C. elegans very easily. I'll explain few manipulations in the next slides. So how we use C. elegans to understand all these biological processes. So we take the worm, we grow, we do manipulations. I'll explain few manipulations. We take the worm, put under the slides, small glass slides, and we check under the microscopes. So there are different kinds of microscopes we use. This one is right field microscope. You just see if we want to do behavior, like say how the worm is moving and all these things, we can just fit a camera with a simple microscope and can detect. But we want to understand how the proteins are moving, from which compartment the protein is moving to the next compartment. We use another technique, which is fluorescent microscopes. So fluorescent microscopes, so a very basic example. So you must have seen in your childhood that there are watches, when you shine the light, it glows for some time. Similar kind of process, not the same, but similar kind of things we do to check the neuron, to check the proteins. What we do is we tag a protein with a fluorophore. So if we shine a light, it will give a different color of light. So if you want to check the proteins, which two proteins are going together, what can we do is we tag a protein A with a fluorophore, which gives, let's say, green color. We can tag another protein which with the red color. So when we shine the light, we can see both two different proteins. If we can see the green color, so we can say the protein is now one protein. If we see the protein in the red color, it is another protein. So this is the one way we check. We tag the protein and see under. Also, there are lot of there fluorescent proteins. They are also the proteins. So when we shine the light, there is a phenomenon called. Okay. So we do by making transgenic lines. So we know a sequence of our protein of interest. Let's say SNP1 is the one protein. What we do is in the same DNA, we put EGFP sequence also. So when DNA, now the SNP1 sequence, just after that EGFP sequence is there. So after what will happen is mRNA is from DNA, mRNA is formed. And when mRNA is translated to form a protein, now the SNP1 is tagged with EGF. So now the SNP1 is attached with. So we can put the dye also. We can, but make a protein. Yes. Which have these fluorescent. Yes.
How is it that you can freeze this C. elegans worm and get it back to life and these experiments? You cryopreserve.
Yes.
How many cells survive?
How the cells survive? So there are some. So we use different materials, different solutions where the cells can cryopreserved. Exact mechanism, I'm not sure. I'll get back to him. How specifically in case of C. elegans it is happening. We'll get back. So we can inject the protein, but in this case we are not injecting the protein. What we are injecting is the DNA. So DNA has a protein. So DNA has a sequence for the protein of interest. In my case, let's say protein 1, I'm checking SNP1, along with the signal, along with the sequence with the fluorescent protein. Oh, so what we are doing is we are injecting in the gonads, So it forms an array-like structure. So there are, so you inject in form of a plasmid. There it forms an array. It can act as an extra chromosome. The C. elegans has 5 chromosomes, but it can either integrate into the genome of the worm also. Physically injecting, we inject in the gonads. What is happening in the gonad, we can. No. We just take the plasmid at different defined concentration. We inject into the gonads. Where, yes, needle. Next generation of the worm will have. It will stably integrated or not, that is a different question. So we can actually CRISPR modify also. We can put, so this is extra chromosomal. Let's say we are putting extra plasmid. But right now what we can do also is itself in the genome of the C. elegans, we can replace a sequence, put EGFP after the protein. So we can do that also. But mode of delivery is by gonadal injection only. Yeah. Yes. So we. Yeah, it's hard. Okay. So we made an agarose pad of defined percentage. So if we stick the worm, one worm will stick. And if we dry the agarose, so what will happen is worm is moving if the surface is wet. So we dry that agarose pad, so worm will stick to that pad and it will not move for some time. So we should be fast enough to inject so that worm will not desiccate. So worm is now stable. We align the gonad in such a way a needle comes and poke to the worm cavity, enters into the gonad, releases very small content of DNA and come back. And then we rescue these worms into a different place. Right now, I don't have. I'm not put in. Yes, a very small needle. Yes. So you need to learn. So it takes some time, at least 1 month, to learn all these making transgenic lines. It's not easy. That is. No, it doesn't die. If somebody know how to do injection, worm doesn't die. So we. Yes. So sir. No. So we don't. I mean, we are not. So everything is stable. Like microscope setup is there. What we have is needle is fixed. We just so needle position is fixed. Now we have to change the stage. So worm is present on a stage which we can move. We can take this. So now worm is like, let's say this is a worm. This is a stage. We search like this is a point needle. It comes into a very close proximity. Slightly we penetrate inside the worm, and we have a controller with the legs. We just press this. Whatever is the content in the needle, it just enter into the gonads. So there are we don't do sequencing, but there are different ways. One is Sanger sequencing. So we send it to some company, and then they sequence the DNA for us. But there are different methods you can read online, Sanger sequencing and all, where they fluorescently tag different nucleotides. So one way is to do it. You have a DNA sequence. Now you are doing PCR polymerizing. You are attached different fluorescent, different labeled nucleotides. On the basis of nucleotides, you see what is the sequence. But there are different ways. There are very fast, high throughput sequencing also. You can check that here. Okay. Okay. So yeah. So what we are interested in understanding is the biogenesis of PSVs. So I spoke about all these Golgi, everything where the proteins are formed. These processes are happening in the cell body. So we are interested in how these vesicles are forming from in the cell body in different compartments, how the motor is attaching with these compartments, and when you order a parcel, what will happen is they use some truck or something like to check to take your parcel towards your home. Similar kind of things happening in the cell also. Cells have a molecular motors. We have the motors like truck and other things, but cells have a small molecular motors. They run on the small tracks also. These are microtubules. The precise name is these tracks are microtubules. But these vesicles interact with the motors and reaches to the synapse. So we are interested in understanding all these things. So this is one example of a video where we tag one synaptic vesicle protein with a green color fluorescent protein, GFP. We tag another synaptic vesicle protein with a red fluorophore. What we are doing is we are providing a laser which excites GFP also. We are providing another laser which excites M-scarlet also. So EGFP is a fluorescent protein, M-scarlet is also a fluorescent protein. You can see on the basis of color, one gives the green color, another gives the red color. So after excitation, we can simultaneously, on the same point of time, we can see both the colors, both the vesicles. So you can see this is the cell body. It's a long axon. So we are using C. elegans PLM neuron. It's a long neuron. And you can see there are certain vesicles which have only the green. There are certain vesicles which have only the red. And when the vesicles is moving, when the vesicles have both the proteins, the vesicles you can see in the yellow color, because now green and red will form a yellow color. So these kind of things we do in our lab. So what we know about the biogenesis of, so this is a published work from our lab. What we know about the biogenesis of synaptic vesicle proteins, or these precisely synaptic vesicles. What happens in the cell is, so you have a compartment from where the vesicles are forming. They have a lot of proteins. So first thing you need to do is you need to exclude the other proteins from the synaptic vesicle protein. So first thing you need to exclude. So here, exclusion of non-synaptic vesicle proteins from a synaptic vesicle protein. So let's say this is the one compartment, let's say Golgi. It has a lot of protein, some Golgi proteins, some dendritic proteins, some synaptic vesicle proteins, and you only want synaptic vesicle proteins to come out. What you will do is you exclude these Golgi proteins from a compartment. So now the exclusion is happening. The compartment only has synaptic vesicle proteins, and then vesicles can form. How it can form is one way I've already explained, that it is recognized by the cargo adapters. So another step can happen is the inclusion. So cell has different proteins. Your synaptic, for a synaptic vesicle, they are different sets of protein. What you need to do is you include all the synaptic vesicle proteins into a common compartment. So there are two steps. One is exclusion. You don't want other proteins, so you exclude. You want same synaptic vesicle protein into a common compartment, so you include into a compartment. So here, these two different synaptic vesicle proteins comes into a common compartment, and it moves integrally. Both are. So that's why inclusion is happening. So initially here is the only one compartment. It can break off, but what you want is when you need a vesicle to function. Let's say synaptic vesicle has a composition, and the vesicle will not function if it does not have all the sets of protein. Let's say if you are only carrying a protein which helps in the fusion, but it does not have a protein which helps in filling of vesicle. So the vesicle is of no use. So you want a right set of proteins for a vesicle to function. Which? No, this process is, as a process, this is not random. There are regulators involved. Like, so fusion is also, you require specific sets of protein to fuse with the compartment. For biogenesis also, you require specific cargo adapters to form. What triggers, if you ask initially from what is happening, that we don't know. As a process at a single vesicle, what happens is, and go back here. So in a compartment, lots of proteins are present. What happens is these cargo adapters comes and interact. After this, it starts building up. There are lots of now more cargo adapters are coming. After this, some changes on the membrane curvature. Membrane curvature is changing. So I have not mentioned in the slide, but there are some other proteins, dynamin kind of proteins, so which sense this curvature of a membrane. Now they sense when the membrane is curvature, and these proteins help in pinching of vesicles from a compartment. So this process is tightly regulated. So we cannot say if all the proteins are present in these kind of Brownian motion in a compartment, because these proteins interact with some lipids also. Let's say you have a membrane, and membrane may not be the uniform. There are certain proteins which interact with some kind of lipid, let's say PIP4 or PIP3. There are different lipids. So there are some proteins which tends to present in the high cholesterol regions also. So they are not completely flowing. They have some restricted zones where they can present. Okay. So so what happens? So we understand about the sorting signals. We know how the proteins are targeted to one compartment to the next compartment. Now, what if the protein is mistargeted? Or what happens if we perturb the machinery? If we remove the machinery, something in any way, what happens to the protein? So let's say I'm just giving two different examples. These are the C. elegans head neuron. This neuron is ASI. So you can see this ring-like structure. This is an axon of a neuron. This long straight tube. So this long straight, or you can use any example, but this long straight tube is the dendrites. So I have already explained that neurons are polarized cells. They have a defined axon and they have defined dendrites. So this round structure is axon, this long is dendrites. So in the wild type, where wild type means the worm does not have any kind of perturbations. Synaptic vesicle, इसमें one synaptic vesicle protein. And synaptic vesicle protein should have, where should they present? They should present in axons, because they connect to the next cells. In the wild type, I'm not sure if it's visible from far, but these SNP1 synaptic vesicle proteins are only present in this ring-like structure. We can turn off the light actually. Is it better? Okay. Okay. You can see you can use this diagram actually. So it is a one cell, actually two cell. So this round-like structure is an axon. Oh, it's in worm. It is in the worm, head of the C. elegans. Head of the worm. So this is a neuron. We only express, not we, whoever done this experiment, they only express this SNP1 GFP only in this particular neuron. So we can regulate this also, where we want to express the protein. We don't want to flood the whole animal with this fluorescent protein. So we use different promoters, different sequences of DNA, which express only a certain kind of neuron. So what they have done is they express SNP1 in ASI neuron. Now coming to the structure of ASI neuron, this ring-like structure is the axon, and this long structure is the dendrite. In wild type condition, what is happening? SNP1, which is a synaptic vesicle protein, present in the axon only. You can see this green color, and it is absent in dendrites. In wild type, in normal condition. If we remove a machinery, you don't need to remember the name of this gene, but LRK has a role in biogenesis of these vesicles. If you remove this machinery, or the gene which is present in is now down-regulated, now the protein is not functional, or protein is not there. So there are different ways we can perturb. But let's say if this gene is not protein is not there, now you can see the protein is present in the dendrites also. So synaptic vesicle in normal condition, they are only present in the axon. We remove the machinery, now they are mistargeted to the dendrites also. They check with another synaptic vesicle protein just to prove, because mistrafficking or mistransport of one protein does not necessarily mean that whole synaptic vesicle proteins are mistransport. So they check another synaptic vesicle protein, SNP1, it is also mistransported to the dendrites. You can see small dots here, which are present in the mutants. Mutant means the protein is not functional here. In that case, in wild type, it is absent.
What these guys have done, they took a protein that is supposed to tell the synaptic vesicle where to go.
Where the protein. So if they tag
protein, and the synaptic vesicle will in the wrong direction as well.
In the wrong direction, you can say that, yes. They are going in the wrong direction. If you remove this machinery or this regulation. So this is cell biologically. I'm going to explain what happens to the organism level in the next slide. So another experiment, this experiment done in our lab, we have used another neuron in the heads, AWC neuron. No need to remember the name. Similar, this ring-like structure is axon, long structure is dendrite. Okay. Now we tag a dendritic protein with a GFP marker. So now we can only see the this order 10 is a dendritic protein. In wild type, it is not present in the axon. You can see this is not present in this ring-like structure, but it is present throughout this process, right? Now, if we remove the machinery, which is AP3 is one of a adapter proteins, which I was talking about. It helps in sorting. So if we remove this APB3, now the proteins are flooded in the axon also. So this machinery is very important for targeting of proteins to a different compartment as well as different regions. These processes, this merge targeting may or may not have physiological significance, so they may not have causes any disease. So I'm going to talk about one particular diseases which see in humans. What if the protein is mistargeted? So in wild type condition, what happens is proteins are formed. In a table I showed there are certain proteins which have mannose-6 phosphate receptors. If the protein has mannose-6 phosphate receptors, they goes to the lysosome. And what is the function of lysosome? To degrade all other proteins which are not required in the cell. Clear? So this mannose-6 phosphate receptor is required to target the protein to the lysosome. Now, if we perturb this machinery anyhow, what will happen is now the lysosomal proteins will not enter into the lysosome. Now, what is the function of lysosome? Lysosome function is to degrade the proteins and other compounds in the lysosome, which are non-required by the cell. What will happen? In the lysosome, these enzymes are not present which needs to digest. In that case, the lysosome will not function properly. If the lysosome is not function properly, cell will have a lot of other things, right? Cell will flooded with other things we don't want in the cell. What will happen is this machinery perturbed, cell have other proteins which are accumulating inside, and this is one disease which is Batten disease. This is caused by a mutation in a gene CLN3. In that particular case, you have mistargeted of the lysosomal protein. What will happen for the humans? It's a rare genetic disorder where from starting of 2 to 3 years, they start seeing the loss of vision. They cannot see things properly. After that, when they start growing, they start seeing that behavior and learning problem. They can't learn. And as they slightly go older, they have the motor skills. They cannot walk properly. They can not use their limbs properly. So this is one way of, this is one example. If you perturb the machinery or in any way, there is some mutation in which this machinery which needs to sort a protein is not functioning properly. So you can have some kind of diseases. There are lots of diseases associated with mistargeting or of these different proteins. Let's say neurons. In neurons, a lot of neurodegenerative diseases just because of the lysosomes are not functional properly. So next question is why it is important to study protein trafficking. Why government is spending a lot of crore of rupees to just funding our researches? Why we are spending our time to understand all these processes? Is it important or worth to look at? So I'm going to ask a question. Let's say you are an engineer and your job is to you're a mechanical engineer, and then your job is to repair a factory. Somebody called you and say, my factory is not functioning properly. We cannot get the end products. Please come and see what is the problem. So they will not call anyone. First, they will call the person who know how that machinery works, right? How the factory works. So to understand, first you need to understand how factory works for an engineer. Same thing, it is very important to understand how these processes happens in the cell. If you know how these processes happens in the cell, and if a patient comes, you can know what is the disease mechanism. If we go back to a previous example, if I don't know how these mannose-6 phosphate receptors and these machinery helps in targeting a protein to a lysosome, I will never understand what is happening in this particular mutant. So the first thing is to understand the cell function. If I know the cell function properly, then I can understand the disease mechanism. Now, let's say you are an engineer, you know how the factory works. Now you nail down what is the problem in the factory. Next thing, what you can do is you are skilled enough to find out how can I increase the production or how can I repair the factory or machine. Same thing we can do also. If we know both things, how the cell normally works, what went wrong in the disease condition, now we can target, we can do some manipulations, we can change something, and we can design some drugs or therapeutic studies to treat this problem. Okay. So how much time I have? Oh, okay. So I'm going to give one example of this, why it is important to study protein trafficking. So Alzheimer is a disease. It's a neurodegenerative disease, and we see this in the old patients. So as the age progresses, the symptoms of disease you can see. So first, starting with dementia, people forget what they are doing, what their daily task is. Even they don't remember what 5 or 10 minutes before they was thinking or doing. So they start with this basic dementia. And then after this, they will start showing like some cognitive disorders, like they cannot solve the problems, they cannot understand someone what they're saying. After some time, they are starting sleep difficulties. And when in the later stage of diseases, they cannot even do their daily functions. They forget to eat, they forget to do other daily tasks. So they continuously need assistant to do their daily function. So it's very, very much prevalent in USA. There are approximately 50 million of patients in the US. So it's very common neurodegenerative disease. How it happens, exactly we don't know. Exact pinpoint we don't know. But it is associated with the two things. One is the accumulation of this tau neutrophils in the cell, or this amyloid beta plaques in the neurons. They affect the function of a neuron. I'm not going into the details how they do that, but they affect the function of a neuron, and the neurons start degenerating. So in the brain, now you are losing your neurons as you age. Now you are losing your neurons. What will happen is neurons function is to regulate other thinking process. Some neurons have a function to walking and other different functions. So now on the basis of which neurons it is affected, you are seeing the symptoms. So this is a summary of Alzheimer's disease symptoms and how it can happen. Now, there are different ways there are different conditions where you can see the Alzheimer's diseases. There are different conditions. Out of this, one condition which already we know is retromer complex. So you don't need to remember the names, but that function, this retromer complex function is to transport these proteins from endosomes to Golgi. It's a basic. We know that already. In the role of this whole retromer complex is to transport the proteins from endosomes to Golgi. What happens in some cases of Alzheimer's diseases, that complex is now not functioning properly. If this complex is now not functioning properly, what will happen is, due to which there is an increase in accumulation of these A beta plaques in the neurons. Clear? You know the normal function. You already know that there are certain mutations or there are certain problems in this retromer complex, due to which you see these kind of plaques in the neurons. Now you already know. What if I design a drug which increase the level of this retromer complex, either, or it increase the stability? Let's say the retromer complex is now hyperactive. It's more functioning. Disease condition, it was underperforming due to various reasons. We can discuss it later. But due to certain reasons, it is underperforming. Now we use a drug which allow us to increase the function of this whole complex machinery. You can think for it like this can be one potential treatment. This is same happening in this particular drug. We use R55 drug. So this is these data is from neurons and culture, slightly complex, but we'll understand. When they vehicle means they are not using a drug to treat. Okay. When they use a drug, VPS 35 protein level is increased. It's a part of a complex. Clear? Another complex, VPS 26, it's also a part of complex, its level also increase. So giving a drug to a cultured neurons, we are not using the human neurons till now. The drug is not in the clinical trial phase, but administration of drugs to this cultured neurons, you can see here these retromer complex level of protein is increased, or we correlate with let's say this endosomal retromer complex is now more stable. Now it is functioning. And when they check this flex in the cell, they see that it is reduced. So this can be a one potential treatment for Alzheimer diseases. This is one. There are different VSS, which are, this is not still now in the clinical trials, but there are different monoclonal antibodies. What they do is they produce antibodies against, small antibodies against these amyloid beta fibrils. And then after binding with this, now their protein, now these this complex of antibodies with amyloid beta, they start digesting it, they transport it to the lysosomes and all other functioning. So this is one potential treatment. Yeah. So have any doubts? You can ask.
Okay, so the topic for questions. After the video, what we can do is we can see some online questions.
Yeah.
And we take them first, and then we shut down the online thing, and we can continue. Online, direct. But are there any indirect questions that we something that is worth talking about? So there was one question on freezing cells. How come you can freeze? You said at some point you can freeze cell.
No, C. elegans. We can freeze C. elegans. Yes, at different stages, L1 stages only. At larval stage 1.
Okay, whatever that stage, you can freeze the worm.
Yes.
So somebody asked, how do you freeze a worm and again make it alive or whatever, or unfrozen? And the next question was obviously, why can't you then freeze a human and make them again alive after a few years or something like that? So yeah, I thought they were interesting question. I don't know why any answer to that, but still.
Okay, so maybe I'm not sure about, I don't know the exact answer for humans, but there are a lot of, so humans have different processes.
I mean, if I freeze a cell, I mean, when I take, I don't know, if I freeze.
Cancer cells, we do freeze a lot of cancer cells.
Forget cell. If I take some bhaji substance,
Yes.
I can freeze it.
Yes.
When I take it out, because the water in the things has become ice and burst, it becomes very soggy.
Soggy, yes.
So take it out of the freezer. So how come your worm survives?
So we use glycerol, different kind of, one glycerol, which helps in the cryopreservation of these worms. Like if we freeze worms with the water, they will not survive. They will not come back. So we use glycerol solutions for that. Busting, yeah.
So why does it see it's burst when you freeze it? You add certain
chemicals to that area.
So I suppose then the answer is a single cell or a very small thing could possibly freeze and thaw, but human being that is now we cannot freeze them.
Yes, we cannot.
Can I do something to make my vegetables also not? This is some liquid I put it and then freeze or something.
You can do, but maybe that the vegetables may not remain edible for use.
Okay, that's poisonous, so it doesn't.
Yeah.
That is okay. Any other questions?
Why there are some reports that indicate a decrease in that is my answer. With certain cognitive games like playing word games can prevent Alzheimer's. Is that true?
I'm not sure about that. Or early onset, it doesn't. Okay, yeah.
So there are no more questions online. Okay. Then let me just remind the online viewers that the next GIY that is on 31st of March, the 5th Sunday. We will be live. It's an online only session. We will be live from the low temperature facility in PIFR, where we will show you how they make different cryogenic liquids and maybe freeze things with it, but not worms. That's the next GIY. And after that, the first Sunday of April is going to be on identifying molecules within living cells, in particular fats or lipids. How does one? It's a sort of chemistry, biochemistry thing where they are designing sensors which can function in live cells and identify very specific fat molecules and where they go inside the cell. So that's coming up in the first Sunday of April. Those are the next two, and we will let you know the schedule after that in a bit. So on that note, if there are no further online questions, we can shut down the online session.
Yeah.
Online people do not get chai, and then we will have chai and continue over here.
Yeah, that's the plan.
That's the plan. So are you done? Then you can Then you can switch off the. You can also
Okay.
unshare and Which I will.